the Dementia Researcher Podcast, talking careers, research, conference highlights, and so much more.
Hello and welcome to the Dementia Researcher Podcast. Today's episode is coming to you from the ADPD conference here in the beautiful city of Copenhagen, where researchers from around the world have gathered to share new findings on Alzheimer's disease, Parkinson's disease, and other related neurodegenerative conditions. Hello, I'm Professor Dave Cash from the University College of London, and I'm delighted to be hosting today's show. ADPD is one of the major international meetings in our field, bringing together researchers working on everything from basic molecular mechanisms to clinical trials, neuroimaging, and new diagnostic approaches. Over the past couple of days, there have been hundreds of talks and posters. So rather than trying to summarize everything, today we're going to focus on a few highlights that really stood out. Joining me are three people who have been exploring the conference: Grace Thompson, a PhD student from the University of Exeter, Dr. Marieta Vassileva from the University College London, and Alice Carstairs from the Alzheimer Society. Hello, everybody.
Hello.
Before we begin, could I ask each of you to briefly introduce yourself and tell listeners what areas of research and work you're in? So we'll start with you, Grace.
Hello, I'm Grace Thompson. I'm a PhD student at the University of Exeter, and I primarily research the role of microRNAs and small non-coding RNAs in Lewy body dementias.
Great, thank you. And Marieta?
Hello, thanks for having me today. I'm Marieta, and I'm a research fellow at the UCL Alzheimer's Research UK Drug Discovery Institute, and my background is focusing on microglia and their role in dementia and neuroinflammation in general.
Hiya, I'm Alice. I am not a researcher, but I work in the research communications team at Alzheimer Society. So I get to talk about all of the great kind of research that our funded researchers are doing and disseminate the outcomes and impacts from that work.
For listeners who have not joined us for one of these conference reflection episodes before, here's how it works. Each of our guests will take a turn sharing a talk, a poster, or a session that stood out to them from the conference so far. And then when describing the work, we're asking them to summarize what the research was about, what the key findings were, and why they think it matters. After each summary, we'll have a short discussion before moving on to the next highlight. And we'll go around the room several times, depending on time. So Grace, let's start with you. What's one talk or poster that's really stood out for you for the conference so far?
I really enjoyed yesterday. There was a number of talks that focused on single-cell RNA sequencing. And being in epigenetics myself, it's very interesting to see. But one of the talks that really stood out was a talk from Melissa Grant-Peters at University of Cambridge, where she was looking at the molecular and cellular differences in Parkinson's disease and Parkinson's disease dementia. And it's really important work because approximately 40% of patients develop dementia when they have Parkinson's disease. And her work used single-cell RNA sequencing to look at the different transcript usage in Parkinson's disease and Parkinson's disease dementia. And they actually found quite a difference in Parkinson's and Parkinson's disease dementia, which I found really interesting. With my work being quite similar as well, it's quite nice to have that overlay and touching on work that's not been done before.
Okay, and for those simpler neuroimaging folk of us, give us a little bit more about why you think epigenetics is so important in different forms of dementia.
It's because I think a lot of the literature highlights that there's a role of dysregulation in genes, and I think with the technologies developing like RNA sequencing, small RNA sequencing, we can really capture why certain cell types are more vulnerable than others. And we can really highlight some of the pathways that are being affected that may not have been done traditionally. And I think with some of these new technologies, we're really pushing them to be able to achieve quite a lot of information about what's happening on a cellular and molecular level. And I think that's really important when we go and design future therapeutics or biomarkers.
I think, yeah, I agree. And I think yesterday for me also one of the highlights was that there was a lot of omics work in general. It seems to be like something that is still very highly explored in the field. I do remember that talk, and it was very cool. It's very outside of my area of expertise, but I still thought it was a really nice talk. Yeah, very interesting.
Yeah, the layers of omics, I think it really highlights the information that we can gauge from different technologies.
I was just going to say that I missed this talk and now I'm gutted. I'm going to have to go and watch it back. It sounds super interesting.
That is one of the real benefits now of hybrid conferences nowadays. There's always so much FOMO. I want to go to this talk and this talk are occurring at the same time. And now, at least with on-demand and streaming, you can kind of catch up a little bit.
4 or 5 concurrent sessions.
Exactly. So just going back to that, what were kind of some of the pathways that might have been different between Parkinson's and Parkinson's disease dementia?
So from what she said, it was that there was a differential usage of APP and SNCA transcripts, particularly within excitatory neurons. But it was almost that the Lewy body pathology was actually the driver of the differential transcript usage, which is quite interesting because it's still quite underexplored. So, I think there's still got a lot of work to do. But I think when you add all these layers of transcriptomics, various multiomics, it really does build up a big picture of the pathways that we could target. Yeah,
and it gets to the point of, are we looking at subtypes, different diseases, a spectrum that we're along? And especially when you're mentioning APP, instantly, sort of the Alzheimer's disease person in me, that rings a bell. So it's interesting that, how we frame these things around co-pathologies or spectrums is more of a construct that's helpful for us rather than maybe...
She did mention that in their findings, they were actually seeing that PD and Parkinson's disease dementia are actually more distinct than they were similar, which does vary across the literature in the field. So it's quite nice to be able to actually see some evidence that they might be distinct as well.
Okay, great. Anybody else have any more comments on that talk?
No, I think just highlighting what you said, just using these technologies to kind of start dividing more specifically the diseases, not just generalizing and saying PD, AD, because now we know it's not really that simple, and usually there are co-pathologies at the same time. And I think that came a little bit from, I think, one of the plenary lectures in the afternoon from Leah Greenberg, the pathology, where they look at the different subtypes, and that was really interesting, yeah.
Yeah, absolutely. Co-pathology seems to have been a real theme this year. It's come up in a lot of the sessions and the talks that I've been to. The plenary speaker, she said, Leah, yesterday was absolutely fantastic, sort of looking at the neuropathology and the autopsy and kind of tracking back the kind of different co-pathologies. So yeah, it seems to be a real theme.
I think it was beautiful work where you can see, because she obviously showed some of their research and how they're looking at brain slices, and it's kind of highlighting, I think she said it in her talk as well, that I think histology has been discarded for a while because now we have these new techniques all the omics, but it still has a place, and we have to, in a way, integrate them all to get the best.
For sure. And then what she was saying about relating it back to biomarkers was super interesting to me as well, that actually some of the biomarkers we have can only identify things into the intermediate and late stages. But when you're looking at sort of brain autopsy, there's obviously all of these stages before that we can track with biomarkers. So yeah, it was a super, super interesting talk.
Yeah, Leah Greenberg has been such a fantastic career and just an enthusiastic champion of science and research and a good mentor, I think. I think it really shows the power of neuropathology. We were talking about exactly what you were saying before. People went away from pathology for a little while, and now a lot of the imagers are coming back because they want to know what does this really do? What does this really measure? And we're beginning to see the power that these things are not competitors. They're things that help each other out. And I found it fascinating. A lot of her work was based on the Sao Paulo Brain Bank, which was the largest population. I never even knew there was a thing of a population brain bank before, but this whole idea of a population brain bank and the ability to then bring in epidemiology into pathology. But we kind of digressed. We kind of had a group highlight there on Leah Greenberg's plenary, which I think I've heard from lots of people was really important. And things like the locus coeruleus and the dentaraphe nucleus identified from her pathology work is now an area actively imagers are targeting. Can we make some measures there? What connections are there? How is it being affected during the disease? So it just shows that, virtuous cycle in that regards. But
yeah, because I think it's always nice to see the functional outcome from obviously the Omics tells us a lot about pathways, but it's hard, I think, especially from our perspective as drug discovery researchers, you want to be more specific. If you have to identify a drug target, I think the Omics informs us only so far, and then you have to have something to be able to look at.
That's really important. Yeah, definitely. So, Marieta, I'll turn to you now. What presentation caught your eye, or poster or talk that you've seen so far these first 2 days?
I think for me, I'm going to highlight the whole session that I think I'm very biased towards. So I thought that the session, which was focused on microglia, astroglia, from mechanisms to potential treatments, was very good. And there were a few talks there that I think were a big highlight. Peter George Hislop, Matthew Burton-Jones, Soyeon Hong from UCL. Also pretty interesting, and I think I personally found them very interesting because they relate a lot to my work, but also the kind of range of techniques that was shown there. There was everything from omics to biochemistry, kind of classic IP pull downs in cells, different models like iPSCs, integrating iPSCs with mouse models, which is something that I think we're definitely moving towards in the field. So I thought it was such a wide range of technologies, and then showing very mechanistically, I think each of the speakers in their respective protein of interest, how they've mechanistically looked at the pathway and tried to figure out what specific proteins play part in what mechanisms in microglia. And I thought that was really interesting to look at.
And kind of if you had to say 2 or 3 takeaways from the session, seeing how all those talks kind of fit together, what were kind of the kind of big takeaways that we're getting from the research in the microglia in the field right now in dementia?
I think we're definitely we're gaining more and more insight into how these pathways work, because I think we've known for years now that there is genetic risk associated with microglia. We know that there's key receptors like TREM2, but I think for a while we haven't had that super specific mechanistic understanding of how the pathways work, what protein interacts with what protein downstream, that sort of thing. And I think now through these researchers and their brilliant work, we're starting to understand a lot more about the specific proteins that we can target. For example, in Soyeon's talk, we heard about MFG8, which is a factor that is involved in how astrocytes communicate with microglia, and she's demonstrating that can be a mechanism through which synapses are engulfed, which we know is a key thing that's happening in AD. So I think definitely more and more drug targets.
Well, excellent. I mean, and from an imaging perspective, neuroinflammation is fascinating, because we really feel that we don't have yet a good tool. So we have some various PET tracers, but it's such a complicated, fascinating, sort of reactive process that your brain is doing when it's being, triggered. And in some cases it can be, beneficial. In some cases it can be worse. And our tools right now are quite blunt in that regard. We can only see it's happening. We don't know if it's a good happening or if it's a bad happening.
Yeah, and I think the timing is very key. And I think that came through this morning in Bud Struper's plenary talk, where he very nicely highlighted the different kind of stages of Alzheimer's. He broke it down to 6 different stages, starting really early on from, the initial A-beta oligomer accumulation, all the way down to tau aggregation and cell death. And I think it was really nice how he almost put different time points across it, saying, if you want to treat here, you have to look at this. If you want to treat here, you have to look at this. And I think that's really important, I think, overall for treatments moving forward in the field, and how we even like design trials and what biomarkers we look at, because I think now we know that it's really complex, it's really longitudinal, and I think you can't just measure a beta and say, oh, this is enough.
Curious, did other people attend this session? If you did, what did you think of it?
I didn't. But
No.
it sounds really interesting.
Sorry, I've outed everyone.
Well, that's good. That way, we're providing complementary information, so that's helpful in that regards. All right. Well, great. Alice, I'll turn to you. Yeah, you have an interesting perspective, being a communications officer, thinking about what's important for patients, their families. What kind of research from your perspective, sitting on a funder, really kind of rang out to you as this is something that's important for us to communicate to?
So I think there's been an awful lot of different research that's kind of been highlighted. And I kind of feel like that's one of the nice things about this conference is we almost feel like we've started out in that biomarker and causes and mechanism space. And then later on in the conference, we're moving more towards a kind of treatment space and hearing a little bit more about clinical trials. Really enjoying the range of research that we're getting at ADPD. And I think that's really important for dementia researchers that we continue across that full spectrum. One of the talks that I picked out as particularly interesting came from a PhD student, actually, and that was something I was also really encouraged to hear and see, is how many early career researchers are getting the space to be able to talk and present at these events, which I think is so important. This was a first-year PhD student called Lauren Young, and she's from Tara Spire-Jones's lab. And she was presenting her work on looking at a biomarker for synapses, which I know that, the reduction in synapses is one of the better kind of linkages to cognitive function. So how great would it be if we had a biomarker to kind of be able to link to that? And she just presented some really elegant work looking at SV2A and kind of tracking that and determining that, yes, the changes that you can see in PET imaging with SV2A aren't then linked to changes in protein content, but actually changes in the numbers of synapses. And that was doing some really elegant work with sort of brain tissue cell slices and sort of reconstructing them in a 3D space for microscopy and overcoming some of the technical difficulties with light microscopy and synapses. So I think it's super important that we've got work like Lauren's, which is kind of that early stage, kind of looking at how can we do this all the way through to the later parts in the week when hopefully we're going to hear, a little bit more of a deep dive into some of these clinical trial results.
Mm-hmm. Did anybody else see that, or do you guys tend to work on synapse density or synaptic proteins at all in your work?
I don't tend to, but some of my results are leaning towards that. So
Okay.
it's quite nice to hear that. Yeah.
So I saw that talk on demand actually this morning, and I agree. It was a great talk and a great example again of the pathology working with imaging and really a clear answer to the question. She was really wondering, is it that we're seeing less synapses or we're seeing less proteins in the same amount of synapses? And her conclusion from what she showed seemed to be the former, that we're losing less synapses.
Yeah, and I think it's, being able to take that yes-no question and give a definitive answer is, it's rare in research, let's be honest, but being able to do that and sort of answer those questions so succinctly, she did a fabulous job.
Yeah. And especially it must be daunting as a first-year PhD student to...
I couldn't have done it. There was a lot of people in that room.
So that's interesting. You mentioned some of this might be related to your work. How so?
So my work looks at microRNAs, and some of the results that we're finding is that there's a link with some of the microRNAs that are affected in late-stage Lewy body pathology to synaptic dysfunction. We're still unpicking it. MicroRNAs are pretty tough to work with, but we're hoping to actually implement some more new techniques. So microRNA scope. So this would be probing the microRNAs with an oligo. So you can look at the in situ locations of these. So hopefully, potentially, it might come up with synapses. We'll never know. But in a few months. But it's good to hear that it's kind of leaning towards that in terms of the research as well.
Great. Well, I think if I'm going to take the prerogative of being the host and offer one of my personal highlights. You may be surprised it's about imaging. One of the things I'm really interested is, I work a lot in sort of the at-risk or the preclinical stage of AD, and thinking about when we can start seeing individuals transitioning from one state to the other. So I think there's a fair amount of evidence from recent papers now that if there's both amyloid and tau present in individuals, the time to cognitive impairment is relatively short. Stama Karayani, who is at the University of Gothenburg, was looking at that. So she works with Michael Scholl and Alexis Moscoso there, and they've collected a lot of amyloid and tau PET data. And she was particularly looking at 3 large cohorts: ADNI, the Harvard Aging Brain Study, and the A4 sort of pre-symptomatic trial using solanezumab. All these are publicly available data sets that are great tools for researchers like me. And what she looked at was particularly cognitively unimpaired people, 400-ish people, who had amyloid and longitudinal tau. And she found that, so one of the things that I've been most curious about is, what is that transition from tau negative to tau positive? When does that happen? And with that data, she was able to show roughly about a third of individuals transition over a 6-year process to go from amyloid positive, tau negative to tau positive. And then once they're at that stage, there's actually quite a fair amount of risk involved. But going a little bit forward, who are those people? And we're still looking at that, but she found that older individuals and E4 carriers were, so things that we wouldn't be surprised by are driving some of that transition. The other thing she looked at is that tau positivity is really that driver to cognitive impairment. So the amount of individuals who went to cognitive impairment were predominantly people who were tau positive. And if you're tau negative, it was some other source. So the amyloid negative and amyloid positive individuals didn't jump right to cognitive impairment. And they did it relatively equal amount. So it was really helpful for us to kind of get better timings, get better windows for when we could potentially treat people in the disease. So that was one I really liked.
Yeah, I think definitely. I think there's more highlight on tau, although I think there is a lot of sessions still on kind of like APOE and things like that. But I think definitely I saw a lot of stuff yesterday about tau, developing models to look at tau, because I think that has been like a problem in the field for a while. And we don't have anything that mimics tau pathology in humans very well in animal models.
Okay, yeah. So we have good animal models for amyloid, say. Yeah, okay.
Oh, yeah.
No, I saw that talk. It was super interesting, and that tau kind of story has come up a few times, and I think has also come up in kind of the treatment world, where people are saying, well, is it that we're going to need multimodal treatments that hit the sort of different parts? And obviously it's fantastic we're starting somewhere with our amyloid treatments, but are we going to need those amyloid treatments and those tau treatments to be having sort of such an impact in people living with dementia?
Yeah, and I think, in some ways, the fact that we now have amyloid disease-modifying therapies allows us to dare to dream about something like a combination therapy. Like, it's hard to talk about that when you don't even have the first step working in individuals. So
yeah, and we wouldn't have been having this conversation however many years ago.
Yeah.
No, and I think some of those combination therapies and things are definitely coming up. I was just before coming here, I was at the session where both Sanofi and Therapeutics were talking about their TREM2 small molecules and the developments on that, which are moving forward, and they've both did in vivo proof of concept studies, so that's really cool to see. So I think hopefully soon we're going to see some other treatments coming in as well.
And I liked how in the debate yesterday it was almost proposed that we should look at how the cancer field do it and whether we have several lines of treatment. Yeah. I think that's really powerful, and I think that will probably be the way that it might go. So
just to fill in for the people, they tried to frame it as a debate late in the afternoon. But really it was it was sort of the interface, I guess, between industry and academia, how they could work together, what frustrations there were there. And I didn't really feel like a tension between the different groups, but they had some really important academics like Cynthia Lemaire. They also had Mark Minton from Eli Lilly. So they had Jeff Kircher from Roche. They had the head of the ADDI, Niranjan Bose. So they had a good mix of people who are all coming at it from different perspectives. Yeah. And you're right, like they did bring up this idea of like we need to up our vernacular game, I guess. Yeah.
But it's exciting times. You kind of know that we're like on the cusp of getting there, which just, hopefully.
Yeah, no, absolutely. And hopefully other forms of dementia will follow suit. Any other highlights that people wanted to raise? Any other talks that we haven't touched on already?
I think we kind of briefly touched on it, but definitely biomarkers. Biomarkers are everywhere. Don't think I've been in a talk where they haven't mentioned biomarkers since yesterday. And I've seen, all ranges of like people doing CSF, blood. I think I saw urine biomarkers in one of the talks. Which is very cool.
And then the integration of AI with them as well. That's been a big theme as well, which has been great. Like, it's exciting to know that could make such a big difference.
Yeah, going back to the biomarkers, I saw a talk on a Singapore cohort. I believe the author's name was Joyce Tang. And it was interesting. So she had your MRI markers, your white matter hyperintensity, your hippocampal volumes. She had a whole panel of both like the new LISA and to look at brain-derived p-tau, which I know a lot of people are really interested in at the moment. And she was like, well, what's the most parsimonious model to predict cognitive decline? And in fact, it didn't involve any PET at all. It involved p-tau 217 to give you the sort of amyloid, a white matter hyperintensity marker to give you some of the vascular elements, and hippocampal volume for neurodegeneration. And, when you think about, a blood test and an MRI, the MRI is still, far more accessible than PET. Not super accessible, but it still is something that sounds like it could be a really beneficial element in terms of diagnosis and predicting where people are going.
Yeah, because I think that it's like basically kind of like two sides of the story. One is to do with diagnosis, prediction, maybe what we were saying about the different types of dementia, actually properly diagnosing people. And then I think the second, from like drug discovery perspective, is something that gives us a better readout in trials where you know that you are actually modulating the pathology in a meaningful way. I think that's something quite important.
And it highlights the fact that, different tools, whether they're blood or CSF or imaging, might be more appropriate in different settings like that.
Yeah, exactly, depending on what your target is doing and what you're expecting to see. And I think, again, going back to what stage you're actually targeting. Obviously, if you're looking at tau 217 quite late, maybe earlier on, you want to measure different things that are more linked to the neuroinflammatory aspect.
I think it's been really interesting as well hearing so many people talk about sort of how we use biomarkers as well. There was a sort of I think they called it a forum chat with quite a number of sort of people working in the field who were just discussing, are we at a stage yet where biomarkers can be used as a confirmatory test? There we go. Is it enough on their own? And just hearing that sort of clinician side and kind of, how primary care practitioners are using them compared to kind of how we as researchers kind of are using them. I think that conversation has been really interesting to listen to, and hearing the different kind of viewpoints about, yes, perhaps we could use them at this point, and other people saying, well, no, that's not how they're being used at the moment. We do need the kind of the imaging, the MRI, the PET at the moment, but wouldn't it be good if? And so, I've really valued kind of hearing all of those discussions and conversations as well.
And I guess kind of going more to that level, we're very biomarker and omics heavy in this group. But I was curious if anybody saw some of the session on the first day led by Jill Livingston and Mia Kivipelto about modifiable risk factors, with a very sort of provocative title, "Is Dementia Preventable?"
No, I missed that one. I know Jill Livingston's work, and with the Lancet kind of report, obviously Alzheimer's Society have helped fund kind of part of that, but no, I wasn't at the session. Was there sort of some key takeaways and stuff to kind of come away with?
Yeah, well, I think so. They showed, the finger study originally done in Finland showed that, this, very carefully, very tailored, very intense, multi-domain intervention is something that will really slow down cognitive decline in individuals. And then Jill Livingston talked about the 14 modifiable risk factors as part of the Lancet Commission, and also highlighted a thing that we throw around the number 45% of things that could, if we could somehow magically like wipe out all the risk factors, that's how many cases we could prevent. And what was interesting is there's this acknowledgement that it's different in different countries, it's changed over time. We've seen a drop in age-related prevalence in Alzheimer's disease, but because the age population is going up, we're still seeing increasing cases. And it's different. In some countries, we're seeing an increase in prevalence. We may see that reversed with as obesity or depression or social isolation kind of creeps back into society. So it was an interesting talk about that element of what is preventable, how is that changing, what evidence do we have of trials working? So there's some evidence that providing hearing aids in certain cases can be really beneficial for cognitive intervention. And there was the recent U.S. Pointer study where they did either a self-guided intervention or a more intense one. Not much difference between the two, but both groups benefited from doing that. So what I find, to link it back to what we all tend to do, can we kind of start to find biological bases of what these things are doing and why they're benefiting people in terms of dementia?
That's fascinating. Did they kind of look into sort of the genetic status of any of these people and whether, those people with sort of the ApoE kind of 4 genotype, was there any sort of indication whether it might work well for them or not work so well for them?
I believe they did. I believe they found it actually benefited the homozygotes more than it did anybody else. I feel like I have to check my notes real quick to make sure I got that right. But yeah, and again, they also had a fair amount of like proteomics. So they were looking at what proteins are potentially being affected by these modifiable risk factors. A lot of synaptic stuff, a lot of inflammation stuff. So kind of lends to that sort of missing piece where we don't really have a good handle on.
I think it is really interesting because that all these things contribute on top of like the genetic risk. And we already know like the genetic risk of Alzheimer's is so complicated anyway with like polygenic risk. And then, on top of that you have things like sleep and your diet and exercise and all that feeds into the story. But I don't think we have a good handle on how exactly it fits into the story yet. Yeah.
We've talked about some of the kind of big research areas that we've seen common themes in terms of co-pathology and biomarkers and things like that, and AI. Just curious, did you find any new technologies or methods that you hadn't seen before that really interested you?
I think you already mentioned it, but I think the new LISA technology coming in, I've heard it across a few talks. So I think that will definitely be something we'll probably keep seeing and more people will use it because I think they have a few human panels and a mouse panel, which is very good for like translational research.
For me, I think it was the new AI co-scientists. I think they're really going to be really exciting. And it was great to learn a bit more about how they've been used. And it's going to be interesting to see how we can apply it to the dementia field as well.
Yeah, I agree. I think that's a great point. The AI co-scientists, that they're very, the creators, both the Google and the CBrain, are very key on this being scientist-led and not just free to let it run amok as it does, which I think is good. And as we get more comfortable and familiar with these AI tools, it is incredible how fast a pace they are going, and how we were going to have a tough time keeping up. We thought the blood biomarker pace was fast. This may be even quicker.
Yeah, but I think it's really good to integrate with all the omics, because I think the omics data sets are like so hard to analyze and it takes so much time. And I think, you can definitely use AI to integrate a lot more data than I think humanly you would be able to.
Yeah, I agree, because I feel like in my own data I've got loads that I want to do, but time-wise, so it's going to be great to know, I can do that. So,
and I think picking out patterns that I think you just won't be able to, whereas it can look through, exactly integrate with publicly available data sets and then just identify things that I think would be really interesting for us.
Yeah, I think overall the idea of, we're getting a little bit more comfortable and transparent with using AI and not feeling like we're cheating somehow. As I think some of the things that they've highlighted is there are 200,000 papers on Alzheimer's disease. And I don't know about you guys, but I feel like every week the pile of papers that I want to read that I can't get to is just really difficult. So figuring out a way to kind of direct some method to kind of handle just the overwhelming amount of scientific literature that's being produced out there.
This is where I think coming to conferences like this is great, because you almost get a quick update on everything that's happening.
Absolutely. Before we finish, I'd like to ask Grace, you and Marieta are both presenting here at ADPD. Do you want to just say a little bit about what you're doing and when?
I'm presenting on Saturday afternoon, and I'm presenting my PhD work on profiling microRNAs in Lewy body dementias. So this is from a cohort where we've extracted microRNAs from the brain from a range of DLB, PDD, PD, and controls, and I'll be sharing my results then.
Oh, excellent. I hope that goes well.
Thank you.
Excellent. And Marieta?
That sounds very cool. So I'll definitely come by. Yes, I am presenting. I have a poster which is going up, I think, tomorrow at lunch, and will be there until Saturday. And this is sharing a project that we work collaboratively with between our site in UCL and one of the other ARUK drug discovery institutes in Cambridge. So it's a collaboration we've had for a couple of years now, and it's looking at one of our GPCR targets, or Microglia, and how we're basically trying to identify small molecules to target that. So if anyone's interested, they should come by.
Excellent. Well, that sounds really interesting. I'll try and stop by myself. Alice, anything from the Alzheimer's Society that you wanted to point out's going on while we're here at ADPD?
Yeah, we've got some of our funded researchers speaking. I believe one of our Dementia Research Leader Fellows, Cara Croft, is speaking on Saturday, speaking about some of her research into tau. So yeah, I'm really excited to catch up with our funded researchers and see what they're presenting.
Well, excellent. Thank you guys so much. Any Copenhagen tips so far? Have you enjoyed the city at all? What have you found interesting about Copenhagen?
I think the food is quite cool. I've been to a few food markets, so that's definitely been a highlight.
Yeah, I feel like I've had a lot of pastries.
Yeah,
Nice.
Just carbohydrates for three days straight.
Yeah, I had a really nice walk down by the river, sort of on the first day. It was, I'm not going to lie, it was a bit grey and rainy, but really nice to kind of walk down sort of the different areas and see some of the architecture. So yeah, that was a bit of a highlight. And
I think this is an important tip for people who are going to their conference the first time. Don't necessarily sit in the auditorium all the time because you feel obligated to. Take a mental health break, go out and see the town that you're out, experience some new things. Interact with researchers as much as hear the research, but to actually talk to people and get a camaraderie. Before we go, I guess based on the first couple of days, any themes or ideas you think really are the field is shaping the field right now? Anything that you think is really just a big drumbeat in terms of the conference?
I think we've already mentioned it, but the co-pathologies, I think, keeps coming up, but also, I guess, the heterogeneity of both disease, but also people. We quite often sort of put people into people with dementia and people without, and actually both of those cohorts of people are so different. So I think that's been a real kind of theme and recognition for me.
And I think I'm going to mention it again, but biomarkers, I think that's just everywhere. But I think there's like a really good sense of like, I think the whole field is very positive because I think the breakthrough with the anti-amyloid antibodies, I think that's really changed the way we think about things now. And we're like, okay, now we're in an era where there is some sort of a disease-modifying therapy. So yeah, how can we make this better? How can we move forward, make combination therapies? What are the things that we should be targeting that we're not already? So I think definitely like a very hopeful kind of sense around the whole conference.
Yeah, I think everyone seems really excited in the fields, which is really nice to see.
And definitely, a positive sea change from maybe where we were about 3 or 4 years ago in that regard. So I think it's a good infectious, that enthusiasm.
Yeah, because I think you see things that things are moving forward, and it's not just like negative trial after negative trial after negative trial. Yeah.
Yeah.
Just getting moving.
Okay, that brings us to the end of our first ADPD Conference Reflections episode. Thanks so much, Grace, Marieta, Alice. Thank you for sharing your highlights and insights for the meeting. Really, really interesting conversation that we just had. We'll be recording another episode later at the conference to capture more reflections and emerging themes. There's still, 2-3 more days left of the conference, so lots more research for people to see. And if you want to learn more about the research we discussed today, you can find links and further information in the show notes. On our YouTube channel, you'll also find lots of our posters and short recordings, which researchers talk about their posters. But for now, I'm Professor Dave Cash, and you've been listening to the Dementia Researcher Podcast.
The Dementia Researcher Podcast was brought to you by University College London with generous funding from the UK National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, Alzheimer's Association, and Race Against Dementia. Please subscribe, leave us a review, and register on our website for full access to all our great resources. dementiaresearcher.nihr.ac.uk.